Okay, so today I wanted to talk about the mechanisms of genetic diversity for influenza viruses. And so what we're going to talk about here is antigenic drift and antigenic shift. What I have set up for you are my arts and crafts influenza viruses. So I have a human virus, an avian or a bird virus, and then a swine or a pig virus. And what you can see here is that the genetics of these viruses are a little bit different in terms of the colors, but each one has 8 pieces of its single-stranded RNA genome, and each one has these surface proteins. There's different types of surface proteins, but you could envision these as the hemagglutinin and the neuraminidase, the H and the N. So let's assume we're talking about type A influenza viruses. What normally happens is you will have an influenza virus that will invade a cell, which is a recycled yogurt cup, and the virus will replicate and it will make more virions, baby virions that are just like itself. We've seen this before. This is normal viral replication. But just like all cells, occasionally when it is undergoing replication of the genome, it'll acquire some point mutations. A single base pair will change. When that happens, you end up with largely the same virus, but now you can see by my different colors here on this one piece of the genome, I've had some mutations that we've acquired. And let's say that those mutations are in one of the genes for one of the surface proteins. So as you can see here, I have a slight variation. It's still the same basic surface protein that I had, but it's a little bit different. This is antigenic drift. Normal, slow, random mutations that over time, if they accumulate, can lead to influenza viruses that people don't have great immunity to. You don't know by looking at this whether somebody would have immunity to it or not, but you do know that it's not quite the same virus that we saw before. Okay, and so both type A and type B influenza viruses undergo antigenic drift commonly. Antigenic shift, on the other hand, happens when we have more than one influenza virus infect a single cell. Now that's unlikely to happen in humans. A human would have to be infected by more than one influenza virus at the same time. Also unlikely to happen in birds, again, would have to be susceptible and infected by influenza viruses at the same time. And there's specificity in the influenza virus receptors, the cells that the influenza virus targets, that prevent lots of variety in terms of what can infect humans and what can infect birds. Pigs, on the other hand, have an excellent diversity in their influenza receptors. And so what that means is that pigs, let's assume this is now a pig cell, can be infected by pig influenza viruses, by bird influenza viruses, and by human influenza viruses. What happens here, as you can see, is that we have lots of different pieces of influenza viruses being made all in the same cell at the same time. And in terms of when those pieces of genetic information get packaged up, there's a little bit of randomness to it. And so what you could end up with coming out of this cell is you could end up with a virus that is largely human in terms of most of its genetics. It is capable of infecting humans, but because of that reassortment process, it has also managed to pick up some pieces of pig and bird viruses. And depending on which pieces you pick up, in this case, what you end up with is something like very much like what is thought to have happened in 1918 or in 2009 with H1N1 viruses. We now have a virus that is fully capable of infecting humans, but now has a surface protein and possibly some other elements that we have never seen before in terms of human infection. And so nobody has immunity. This is a brand new strain of the influenza virus. So this is called antigenic shift. It's a sudden, random, drastic process that typically only influenza A viruses undergo, because influenza A viruses have a variety of hosts, whereas influenza B viruses really are only thought to infect humans. The reassortment process is where you have all of these pieces of genetic information for different influenza viruses, at least two, infecting the same cell. And when they get packaged up to make new virions, the packaging has some mistakes in it. You end up mixing some of the pieces.